about 1 million cells and their interactions with the local microenvironment and how they interact with each other. So here, example of how heterogeneous and complex this information could be. So here I'm showing you just a simple example of a long slide. And from there, you can see we're highlighting basically 4 different typical classical phenotype. We're ranging from just completely normal lung to a bit of basically an early problem of adenocarcinoma to a very sort of late stage where you have immune destruction and basically hallmarks of the patient not doing very well. So if you look at, in some cases, those changes in those phenotype also correlate with the location of where those phenotypes are, and making people thinking about why they came about, and then maybe there's an evolution even within the slide between them, or it's actually underlying 3D structure of the tumor dictate that they look like this. So there's a load of information to learn and then try to understand from those images. But sort of disappointingly, traditionally those information are largely discarded. Quite often they were just giving a simple, relatively simplistic single feature. For example, people tend to want to measure the ratio between tumor and stroma or some clinical grading. So there's a lot of information to be gained from this. So I wanted to use analogy between just trying to understand the ecology or geography of an environment. So what we're trying to achieve here is to catalog the different appearances and different haplotype, if you like, of tumors in those tremendous amount of information. So we're trying to build a guide. So that basically guided our approach. So we started from a unsupervised sort of a framing of our approach. We're not trying to predict a certain label. We're just trying to really let the method to learn from different diversity of appearance, just histological appearance by itself. So we started the project around about 2021 or so. So around about the same time, self-supervised learning in computer vision just took off. There's a lot of unsupervised learning at very large scale becomes available. So we team up with one of our local pathologist collaborator, who is a lung pathologist, John LeCain, and Aris Sirigos from New York. We shared the view of doing unsupervised representation learning for histology size. So this is a relatively old self-supervised learning approach, which is called Barlow twins. It is not really the state of the art anymore. If you look at pathology foundation model nowadays, it's basically all different approaches. But this particular type of approach we like really very well is because it has very interesting properties that we like. So what you see is that this self-supervised learning approach relies on quite a lot of very heavy augmentation of the color exposure of the images. As a result, what we find out is that those changes in color actually go much beyond what we typically see as a major source of a technical batch effect in histology, which is stain differences between scanners or institutions. So this approach may be less powerful in terms of getting all the features you may get from the images, but it's actually getting features that are quite robust. The approach is relatively simple. You train your encoders through self-supervised training, and then you process all of those tiles. So by the way, those tiles are small chunks of a larger slide. And we do that because the whole slide is too big. You can't really put it into a single GPU card or with a single network. So you have to break them into parts. So that actually gives you quite a lot of power in terms of training models. So depends on the resolution. Nowadays, you're typically getting about from 1,000 tiles to about 20,000 tiles from a typical whole slide images. host line image. If you have a cohort of 1,000 patients, you get easily get more than 1 million tiles to train your models. So at that time, with the way we're thinking about getting those guides or a guide of histology is to do clustering based on those neural network representations. So here you see the example of a UMAP projection of this. So we didn't invent anything new. We just take the approach that was really pioneered in the single cell community, where they kind of have a similar problem. They have loads of cells, and each cell with a larger, quite high dimensional data. And our data is actually sort of behave nicer than them because our data is very nice, where we don't have really sparse signal in our neural network representations, so very, very nicely behaved due to the self-supervised training objective function. So latent clustering behaves really well in this case. So once you do that, and then actually very easy to gain information from a single slide. So here you see example, the tiled image on the left. This is what you get a typical whole slide image. And then you can easily assign those tiles into this centrally identified clusters. So what we do there is to ask our pathologist collaborators to look at random samples for each cluster and ask the question, are they representing a coherent appearance? Or if they do, what do they represent? And then we write down what the meaning of those appearances are. So this gives us immediately a powerful way of describing what's going on in this complex images. So then what we did is just to use compositional vectors to represent each patient with those as a contribution from those clusters. We call them histomorphological phenotype cluster, which is short for HPC. So we tested in lung cancer a very simple task, which is distinguish between lung adenocarcinoma versus lung squamous cell carcinoma. So these are two major subtype of non-small cell lung cancer. They are easily visually can be distinguishable between those two subtypes. And then we use TCGA for training and test on an independent cohort from a collaborator from NYU. Just by using the frequency of those clusters, you can really solve this task really well. And the nice thing about those cluster-based interpretation is that it's very easy to interpret. We can use a very old established method like odds ratio in logistic regression to understand what's going on. And then when we look at those clusters that are linked to different classes, and then it really is able to pick up or keep all those sort of characteristic of phenotypic appearance of LUAT in this case, and those people would expect to see in squamous cell carcinoma. In this case, what we call a solid pattern. We have a lot of sheets of immune cells there. So then we're sort of encouraged with what we find, and we decide to look deeper into the clusters themselves rather than using a classification task to understand what they are. So first thing we checked is the sort of the technical quality of those clusters. We asked two questions: Are they patient specific, or do we really find recurrent appearance across patient populations, or do we have any problem with technical noise from institution? Using TCGA as example, because there you have images coming from about 30 different institutions and quite a large patient population. We're quite happy with the quality that we have. And then we team up with 3 expert pathologists. We're really looking into using lung adenocarcinoma as an example study. The reason that we chose LUAT is because LUAT is an interesting cancer type that is traditionally been recognized with about 6 different classical growth patterns. And then those growth patterns has been recognized to be important to be predictive of patient outcome. So when we look at basically the left area of our UMAP and those clusters, we find that these are actually highly concentrated with those epithelial or basically cancer appearances. And then what we what we find is that all the 6 classical growth patterns are recapitulated in those clusters, and then in some interesting ones, we find further granularity. For example, the solid pattern, which is traditionally recognized to be a quite aggressive pattern. It's not good news for patients to have that kind of appearance in their slides. And we find different subcategories ranging from very cold, not much immune activity going on, all the way to solid patterns full of infiltrating lymphocytes. And then I think the numerical property of the neural network basically reflecting this UMAP is that a different area actually shows quite nice coherence in terms of their appearance. So on the right-hand side, you see these are relatively normal appearance of lung, and in the middle you see sort of the sample started becoming showing a lot of a different level of inflamed phenotypes. So the nice thing about those clusters is they're very easy to sort of test them in terms of zero-shot setting. So what you can easily do is that when you have a new population or a new cohort, you can easily assign those tiles from the new cohort to those clusters. We find those clusters are immediately predictive of progression-free survival. And then what we did is to link them to underlying molecular data. So in TCGA, every single patient has H&E images. They also have a variety of different molecular profile from gene expression, mutation, and the rest of it. And what we found is that if you correlate the cluster frequencies with the activity of those signature, we find a very strong correlation between those clinically poor outcome group of clusters and good outcome cluster and their relationship with the underlying molecular causes. For example, we find the good outcomes are quite often highly correlated with various degrees of immune activities, as highlighted over here. And the poor outcomes are very much associated with signal for proliferation, lots of mutation-related signatures. Yeah.
Between the effect sizes with the outcome. So is it the case that at some point you sort of start to lose the signal? So for example, if you have the size of 256 by 256, or you break it down to 56 by 56, so will it lose the signal to sort of differentiate the outcomes?
That's a good question. So we didn't actually change the size of our patches. But you could imagine that it depends on the size. You capture different type of features in the image. In some cases, it depends on whether that feature, if it's so small, it's a single cell based feature. Sometimes it may have stronger or actually weaker association with the outcome. So we actually use a very high level a view of the image. We used a magnification that is actually quite low. It's 5x magnification. So in each one of our tiles, we can see quite diverse information of tissue architecture there. Yes, so something like 5, close to 5. Okay. And then so the other thing you could do is to use cell-based classification methods like HoverNet to really characterize what's going on into those clusters. And then that basically sort of corroborate what we find with correlations with molecular data is that those good outcome clusters are indeed full with immune cells, whereas poor outcome clusters are sort of depleted of immune activities. So that was a lung cancer. So in the paper, we expanded our study into several cancer types. We didn't try all of them in TCGA. We used sort of a cutoff about 200 patients. Because the reason we do that is because our survival model in the end is a very traditional Cox regression model. So we kind of rely on we're forcing it to have some power in terms of patient sample to get associations. So basically we find similar things where we defined our clusters across cancer type. And so you can imagine that a cluster defined by slice from quite diverse cancer type. And when we do this, our collaborators a bit puzzled a little bit because that's a not very common thing for them to do, to look at histology across organs. But nevertheless, we find interesting associations that is very much true, quite strong across cancer types. So these are the 4 features that we find that is showing quite consistency across different cancer types. Basically, tumor infiltrating lymphocytes, macrophage regulations, proliferations, and TGF-beta response. So the other thing, these are the 4 things that we find keep coming up. Well, it doesn't matter really. It doesn't really matter which cancer type you look at. And this is just a more detailed correlation for more molecular signatures. So we did some benchmarking with the performance in terms of predicting survival, which survival prediction is kind of the #1 task for those type of images. This was done quite a few years ago, so the methods are already quite old. There's the newer pathology foundation model were not included in this, but still, to our surprise, that at that time the state of the art of summarizing slide level information is something called multiple instance learning, which is already established method in machine learning. It's a very potent correlation extractor, I would say. It's able to find anything in this 1,000 tiles or tens of thousand tiles, anything that is remotely associated with the survival label. So you would expect it to perform really well, but actually, as you can see, compared to deep learning based multiple instance learning, a simple clustering and frequency based predictor is quite competitive. So which begs the question about really what are the predictive signal in those large slices? Are they actually exist in the localized tiles, which is really what multiple instance learning is trying to get, or is actually much larger tissue architectures at play here? So since we established this method, we called histomorphological phenotype learning framework called HPL. So we ourselves and our collaborators are applying this to multiple cancer types, really with the goal of trying to establish this guide of a histomorphological landscape across cancer types. And it's relatively easy to do, easy to deploy on very large image cohort. So we look at mesothelioma. Mesothelioma is a very aggressive cancer type and very unfortunately quite common in Glasgow. So we find similar things, very reproducible and robust clusters in mesothelioma. And the interesting thing is that predictive power and also translate from whole sections to biopsies. Our collaborators look at different cancer types, bone cancer, and also in different therapeutic settings. In this case, they look at colorectal cancer with neoadjuvant therapy in terms of predicting neoadjuvant therapy response. Those clusters seems to carry some predictive power there as well. They also look at response to immune checkpoint inhibitors responses, and they also find those clusters can predict them in the case of melanoma. So for us, we were basically puzzled by why clustering frequency is so predictive compared to a deep learning or attention-based approach. So So what we did in the after this gave us confidence of asking the question, what happens if we really scale up this approach? So we started a university spin-out, which is called TileBio. And the goal of this is to really scale up this tissue language model to millions of host line images, really to see if you properly scale this method, what can they actually really deliver new powers in pathology. So I wanted to switch gear to from H&E to spatial proteomics. So in our institute, we quite often generate a lot of spatial proteomics images, and they are quite often generated from a particular study where there's a very clearly defined hypothesis. So these images are quite often available in large number, but sort of doesn't have a very large panel. So you're talking about 6 or 8 panels, but they're really good at solving a hypothesis. But even with that relatively small colors in those images, analyzing those has been the major obstacle for those projects. So even well-established method of cell segmentation and cell phenotyping and analyze neighborhood takes a very long time to do and very difficult to get right. So naturally, we ask the question about what if you do a self-supervised learning for those? And then because those panels are not huge, we can really use a multi-channel CNN really to deal with this with this problem. So we developed this what we call a hybrid representation learning method. So we combine the self-supervised training with traditional cell segmentation-based statistics to basically mimic what we've done with HPL in histology, trying to establish what are the recurrent appearances in those spatial proteomics data. And then because we often have large patient number, and then really to find out the correlations and their with clinical outcome. So this is basically the training data that we have which come from about 1,000 lung adenocarcinoma patients, about 800 colorectal cancer patients, and so we get about 3 cores. I think these are tissue microarray cores, relatively small, but you can sample multiple from patient. And then, so these are the 3 main panels that we have. You can see they are basically vary between 6 and 8. They're not very large, but even though it's not very easy to do image analysis. So this is the performance of HyperStack between. We tested in 3 settings, and then using predicting patient survival as a task. We did cross validation. We did actually train multiple models in this. So the top row is where you only use self-supervised learning, and then the middle row is just traditional segmentation-based analysis and HyperStack, which combines the two. And not very surprisingly, we find combining the two gives a very notable improvement in separating low versus high risk patients. And that benefit seems to be quite robust across different self-supervised learning training regime. So all the way from the very simple, basic contrastive learning to more newer dyno training method and a mask autoencoder and a VecRec, which is a sort of a newer version of Ballo twins. So encouraged about this, we start to look at what are the clusters these representations are enable you to find, and then it's interpretation of this is much, I have to say, it's much harder than H&E, because even our pathologists can't really look at those images and say what they are. So this is where actually the cell segmentation could come with the tile actually helpful. So you know what are the enrichment of different cell types, so that helps them to write down what those phenotypes are. And And then when we try to combine different panels using a very traditional method, what we did is just we get representation from two panels. We just use a non-matrix factorization to combine them. There's no specific reason why we have to use an NMF. You can do other things, even a neural network-based approach, I guess, will all work. And to our surprise, in this particular long cohort, with just simple fusion enable us to find genuinely new phenotypes that are related to patient outcome. So this is a particular component that came out, this NMF-based fusion from the two panels, which is very powerful in terms of separating patients. And then when we look at what those what that fused component mean in terms of actual image, we find this peculiar looking appearance of GLB1. The GLB1 is when our collaborator made those panel. So GLB1 was selected as a cell senescence marker. So they basically use it as with the assumptions that patient with a lot of GLB1 should do really badly in terms of survival. But what we find is that images where those GLB1 are very close to the periphery of the cell, and patient with those phenotype, they actually do better. And then we were a bit of puzzled by this, and then with the help of Gemini and some other biologist collaborator, we realized GLB1 actually also encodes a cell surface protein. And in this case, it's indeed that cell surface protein is more indicators of better outcome. So we have now we've therefore later done a bit more molecular associations, and then some collaborators are follow up with that story. So now hopefully I can convey this information that now you can use self-supervised learning to do very complex images quite well. It doesn't really matter what method you use. You will pretty much get a positive outcome with your project. So we want to get back to H&E. The reason I want to get back to H&E is that if you look at in terms of making a clinical impact, and also just the sheer number of biological information in H&E, and compared to this its more interesting and molecular counterpart, it's a very big gap between the number of images, the number of samples that you have to build your model or to make your studies. Just to put it another way, so if you look at all the public available H&Es, it's about 70,000 slides, and based on the statistics that I mentioned, that you can expect to get about 1 million cells per slide, you can confidently get about 50 billion cells with spatial information intact in those public available H&Es. You don't have to do anything to get those data. And if you compare those foundation model efforts that is currently happening purely from spatial biology assets, so right now at the moment their cell numbers are much lower compared to H&E. So there's still a lot of values in combining this two, but still using H&E as your main prediction deliver vehicle. So we came up with a method to do that. So the use case scenario we imagined is where you have access to a small set of training data where you have matched H&E and some spatial, should it be spatial proteomics, should it be spatial transcriptomics? But you only have that in the training set. But when you want to deploy your trained model, you only have access to H&E. So this is almost like a knowledge distillation case where you're trying to distill information from those high quality or very high content spatial data to a model that can basically accommodate those information and then find the correlation with H&E appearance, and then use that to improve your H&E encoders. So the model is called we call Trident. It's one of the best name we can find. So we try this approach first with traditional contrastive learning alignment. So this is very much the state of the art. Even you want to align to modality, contrastive learning was probably one of the first methods you would try. So Trident is different from contrastive learning by introducing a additional channel in the primary case. In our case, the primary modality is H&E. So what we found when we tried this exercise with contrastive learning is that the learned model quite often has to find a compromise between features that are appearance that can be found in H&E and spatial proteomics, and they're only able to learn the features that are exist in both modalities. If you have any features that is more strongly sort of exist in one modality other than another, contrast learning basically fails for you to capture that. Just want to use this endothelial cell as an example. So this is a appearance of endothelial cells in the adipose tissue appearance. And so we train everything from scratch, so they are comparable. So this is the model we trained ourselves using just H&E alone, using self-supervised training. What you find that is able to identify features. So these are basically Grand Cam activation maps. It's able to identify features like this, the different cells there, but rarely start to put a lot of emphasis on the region that just seems to be empty. And then we trained with contrastive learning, inference. And then when we interpret the trained model, and the model is able to move the attention away from this middle of the image and rightly focusing on the three endothelial cells. And the main reason for that is we have in the training set, we have IF images basically marking where those cells are. So it knows those things they should put attention to. But at the same time, it loses other important structures in H&E. So with Trident, because we have the additional channel, allows the model to basically maintain the information in H&E. So in this case, it shows a much better activation map. Yeah.
So what essentially is actually picking over here? Because you are only training the color, isn't it? But that's a vacuum over there. So what is there in the density?
So this is the adipose tissue where you also have endothelial cells in the middle. So a well-trained model should be like this, right? Yeah, it should recognize adipose tissue architecture. It should also recognize the cells are there. Depends on different contexts. They will have their values. But the point I'm trying to make is that if you do contrastive learning, if you look at the IF cell mask, you really don't have the adipose tissue texture there. You only have 3 cells, right? That's the minimum information you get in the IF channel. And because contrastive learning is trying to push them align together, so your H&E model has to make a compromise, which you get this, right? And then Trident is really trying to mitigate that. So we test this on a public available data set. So this is not really our own secret data set. So they have a single channel as images aligned with H&E with I forgot how many markers. We train it with Trident and test on 10 different tasks. So all those different tasks are H&E only tasks. And then at the bottom, you see the performance with pairing with different markers and then the performance on the downstream tasks. And we try 2 objective functions. One is VegRec in terms of aligning. So this is really a method that we like a lot. The reason we like it is it has very nice variance stabilization property with it. And then the inference is really the traditional canonical contrast learning case. And then using these two objective functions, and we find the trident architecture really can bring about a very common benefits across multiple markers. There are some markers that are notable different drop in performance. Maybe that is really that particular marker is not very good for that task. But that's gives you the hopefully give the idea about where we're going with this. So we develop capabilities of encoders for H&E and also for multiplex. Now what we're trying to do is to bring them together using knowledge distillation to get the benefit of expensive spatial biology profiling with H&E trained model only. So that's the conclusion of my talk. So in terms of take-home message, hopefully I can convince you that morphological signal among tumor is actually very interesting and very complex. And self-supervised learning is very, at this stage, can really robustly identify them and generating very interesting hypotheses. Yeah, it's my acknowledgement slide. Thank you.